Hello, I'm glad to be here today to talk to you about our project related to how we find toxicologically significant compounds using in vitro bioassays and high-resolution mass spectrometry. This project is a joint collaboration between my lab, with Gabby Black taking a key experimental role, and labs that do bioassays, Mike Denison and Chris Vogel's labs, and Guo-Chun He did a lot of the bioassay results that we're going to talk about today. So as you can see from the background of this slide, we're going to be talking about wildfires in residential areas and the impacts that they cause. But before we do that, I'd like to just give an overview of what it is that my lab is up to these days and how it might be of interest to those of you who are watching this today. So my research starts with a focus on chemicals that we put in products, things that we use in our houses and to build our houses and so forth, because we would like to understand how we make better choices about chemicals in our products based on their ultimate environmental impacts. And there are many different ways that these compounds can impact us. They can be disposed of down the sewer for things that are used in laundry products or dishwashing detergents, things like that. They can be disposed of in solid waste at the end of their life. They can accumulate in our houses, for example, in the dust or in the furniture, in the foam. They might be directly applied out of doors, like ant control around your home. They might burn up in a wildfire, as we'll be talking about today. Each of those pathways can enter intervening compartments like wastewater. They can be converted into biogas and biomethane, turned into biosolids or compost, and could be runoff via stormwater. Each of those compounds in each of those media then are subject to possible transformation in various processes that happen in the environment, such as biodegradation, combustion, or other abiotic reactions that happen, for instance, photolysis. And so in the end, when we sample the environment, ambient water, ambient air, ambient soil, what we find is that we're sampling not just the mix of chemicals that we started with, but a complex mixture of things that have been sliced and diced by various environmental reaction processes. And so that's where we're looking for impacts. And what we want to know is, are these compartments, air, water, and soil, are they healthy for people who are exposed to them or who consume them? And there's lots of different ways we could assess this. We could measure the toxicity towards some particular organism, like an aquatic invertebrate, for example, in a sediment test. We might just assess whether they cause particular biological changes like mutagenicity or endocrine disruption. Our goal in our lab in the red box is to identify the compounds that are responsible for that activity and ultimately to try to connect them back to the product choices that we've made in order to give feedback to people who make these products, to tell them that chemical A is a more safe or greener alternative than chemical B. So that's the big picture of what we're trying to do. And I've chosen today to talk about one specific project, but I wanted to provide some examples of other things that we're doing in my lab at the moment. So the next slide just tells us, provides two specific examples of other projects that I won't be talking about today. The one on the left is our Superfund research program. Many of the methods that are talked about today are developed under that program. I'm the deputy director of that program. The director, Bruce Hammock, and I have worked together for a number of years. And it's really a collaboration between engineers, analytical chemists, and people who do mechanistic bioassays and mechanistic biological impact studies. And a lot of this work and workflow that I'm going to be talking about today grew out of that effort. On the right-hand side, I've just highlighted a big project which we're currently engaged in which we're measuring drinking water samples, treated drinking water from 8 different communities in California at multiple time points. In each of those areas, we have 15 households participating, and we're doing comprehensive non-target chemical analysis of drinking water using the kinds of techniques I'm going to be talking about today. Those results are still in process. And in the center, I've just listed some other ongoing projects that I don't really have time to talk about today. So before we get to the specific project, I wanted to try to connect what I'm going to be talking about to those of you who might be watching this webinar. I know that a number of people might be looking for example, business opportunities related to non-target chemical analysis because it's not as widespread as targeted analysis at the moment. And I wanted to draw your attention to the way in which trends and policies, specifically in California, but possibly other places, are colliding to make non-target analysis and bioassays kind of a hot topic. So in California, we had a really large drought in the 2014-2015 period. A lot of our aquifers were drawn down and there were wells that went dry. And so that caused the state to grapple with this idea of sustainable groundwater management. What do we have to do to keep our aquifer sustainable? And what they came up with was this SGMA, Sustainable Groundwater Management Act, which says that you need to make sure that your pumping of water from the aquifer is sustainable. And rather than, your two choices to do that are to curtail use or to increase supply. And a lot of districts are trying to increase their supply by recharging their aquifers with a variety of water sources, including treated wastewater, agricultural return water, stormwater, etc. At the same time, the state of California, another arm of the state of California, the State Water Resources Control Board, has adopted a water quality control policy, which says, among other things, that you will test these waters that might be used in an aquifer recharge project for not only target compounds like we might expect them to, but also to look for particular in vitro bioassay results like estrogen receptor agonism and aryl hydrocarbon receptor agonism. And so that's a water-based example of the kinds of detective work we're going to be talking about today. So this wildfire study grew out of the firestorm in 2017 in California. And in the aftermath of those fires, there were a lot of questions about how safe it was to go back in and effectively sift through these ashes. We know that obviously a home fire is one thing, but an entire neighborhood burning down at very high temperature in a wildfire is another. And it wasn't clear that when you burn down the households that are constructed from all these different synthetic materials and containing things like pesticides and solvents and electrical insulation and so forth, that all of those compounds might not end up in the ash and pose an extra risk to people who are trying to, workers who are trying to renovate the site or people who are trying to reoccupy the site. So our goal was to understand whether the ash from these residential wildfires was intrinsically more harmful than the ash that might be derived from a normal forest-based wildfire. So to answer that question, we designed a study in which we took samples ranging from the upper part of the map in the center shows Robert Louis Stevenson State Park. That serves as our undeveloped reference site in this case. It was the northern boundary of this particular piece of the fire, and the prevailing winds during this fire were largely from the northeast to the southwest, so angling down to the left in the map there. And so there's a transect of sites. At each general location, we took 3 samples. So there's a total of 5 sites, 4 of them developed, 1 of them undeveloped. At each site, we have 3 different samples, and then we composited samples from these homes. And a couple of representative destroyed homes are shown in photos on the left. And this fire destroyed over 5,600 structures, caused 22 deaths. And we were trying to understand whether the sites in yellow up at the top were really different in their chemical composition and in their biological impacts compared to the urban or residential sites. So what I want to do today is to show you how we do this analysis. We have a unified workflow that applies to both LC and GC high-resolution mass spectrometry data, and then how we connect that to the bioassay results that we obtain, and then to just talk a little bit about the chemical composition difference and whether that matters to what we're trying to say something about, which is toxicity. And as I've already mentioned, we're using these two instrumental platforms, both LC and GC instruments. And we've got these total of 15 sites, 12 of them with residences that were destroyed. So to provide an overview of our analytical method, it's a fairly straightforward and not all that exciting set of sample processing. So we take sieved ash samples from these sites. We do a quick sonication extraction sequenced with hexane acetone and then acetone. We evaporate that, filter it. To do the LC side, we do a solvent exchange. On the GC side, we add an internal standard. There's a series of internal standards added to the LC samples. The analysis is done on two quadrupole time of flight instruments, the Agilent 6530 LC and the Agilent 7200B GC. The LC samples are cleaned up and then run in both ESI positive and ESI negative. And we run them initially in full scan, and then we go back and do targeted MS-MS on priority features. And the priority features are the ones that are related to the bioassay activity, as I'll describe. And our data acquisition on the GC side, we're doing this in electron ionization mode, and we've got both a target screen for some compounds that we knew would be likely important, and also non-target or suspect screening. So I alluded to this unified data analysis workflow, and so here it is. This workflow allows us to take data files that come from our instrument. These are Agilent.d files, and we take them through an analysis-based file converter to make them into a format that can be read by MS-DIAL. MS-DIAL is a program that was initially developed in a collaboration between a Japanese institute and the UCDAVIS West Coast Metabolomics Center. And it aligns and deconvolutes the data. It's designed for metabolomics, but we use it for what you might think of as exposomics. And then after we have aligned features, especially aligned features that are of interest given their relationship to bioassay results, we take those same data and we put them into another program known as MS Finder. And I've provided citations to the development of both of those on this slide if you're interested. So these are freely downloadable programs available through the West Coast Metabolomics Center website and other places as well. And then in the end, we try to annotate these features and understand how they're related to the bioassay results. Recently, we've developed another branch of this process in which we take our Agilent.d files and we use the qualitative analysis programs that Agilent has developed to create chef files, compound exchange format files. And these chef files can be taken directly into Sirius CSI Finger ID, which is an alternative platform for in silico analysis. And we're really enjoying and using that workflow. It's very effective, but I'm not going to be able to talk about it much today because I've got too much to cover. But it's important to know that especially once you get to this in silico MS-MS analysis stage for non-target, it's important to use multiple platforms and cross-compare the results that you get. And as you'll see, we're still working on that piece of this particular puzzle. So why do we use this workflow instead of the workflows that are available through Agilent or through other open source programs? And the short answer is that because we're using both LC and GC data and we want to merge those two data streams, having a single programming approach, which allows us to process both sets of data in a consistent way, is a huge advantage for us. And that's really the fundamental thing behind this, because the MS dial allows us to treat those two data sets more or less equivalently. And as you'll see, we've had some success in both of those directions in terms of identification and elucidation. And the other thing that we've noticed consistently is that when we're talking about non-targets, assigning the molecular formula to a candidate structure using both MS1 and MS2 data is much more robust and much more likely to yield an unambiguously correct molecular formula based on our testing with known compounds that we pretend that we don't know and verify the molecular formula assignment. So a brief 2 slides about the bioassays that we're going to be talking about. The first set of bioassays are referred to as chemically activated luciferase bioassays. They're done in something like a 96-well microplate, and you've got cells in those microplates that have either aryl hydrocarbon receptor, estrogen receptor, or androgen receptor. These are the 3 CALUX assays that are the subject of this talk. And what happens is when there is an agonist, that is something which binds to that receptor, the cells have been engineered to express luciferase and make light, as you'll see in the bottom right panel. And so we then see a light intensity as a measure of how much binding there is. So agonism is when the chemical extract evinces more activity than it had in the control. And sometimes we see the opposite. We see antagonism, where the response is actually suppressed. And that can also happen in a case where the chemical blocks the binding of something to the receptor, for instance, estradiol to the estrogen receptor or testosterone to the androgen receptor, and it actually reduces the activity, and that's referred to as antagonism. And we'll see some of both of those effects in the data. These assays are all done by Guo-Chun He in Mike Denison's lab here at UCDAVIS. The second cellular model that we're working with is out of Chris Vogel's lab. It's a respiratory macrophage model, and the idea is that chemicals, once again, it's mediated by this aryl hydrocarbon receptor, but then what we're looking for are genetic markers in either increase or decrease in the expression of IL-8, COX-2, or CYP1A1. These are markers of inflammation, apoptosis, metabolism, oxidative stress, and other things that might be problematic. Notice that all of these things could be normal physiological responses potentially, but they are expressing a significant change above baseline and in the presence of an active compound. So both of these kinds of assays, we think of them as being necessary but not sufficient conditions for toxicity. If things bind to the aryl hydrocarbon receptor, there is a chance that they're toxic in the same way that PCBs and dioxins are, or there's a chance that they would normally be metabolized by the system and wouldn't be of toxic significance. That's just one example of how they might not be of toxic significance. So I'm going to go through 3 slides summarizing the 6 different bioassays. We have 3 macrophage bioassays and 3 CALUX style bioassays. And each of these 3 slides is configured the same way. The 12 residential sites are shown there. You'll see that there's these 4 neighborhoods: Cough, Lark, MWS, and TBS. And then there's the 1 undeveloped site, which is from that state park, Robert Louis Stevenson Park. And then on the far right is the positive control, and on the far left is the negative control. And the stars, the asterisks on this plot, indicate statistically significant difference from the negative control. And so what we see is that first, there's no systematic difference between the undeveloped sites and the residential sites. If anything, the most active sample is one of our undeveloped sites. but there are several of the residential sites that have elevated activity, both with respect to cytochrome P450 and with respect to aryl hydrocarbon receptor agonism. There are far fewer statistically significant effects in the IL-8 and COX-2 assays, but there were a couple of sites, and the same sites that we saw previously as being highly elevated for AhR and CYP1A1. And then the third pair is the estrogen receptor and androgen receptor assays. And in this slide, the positive control is not shown because, as you'll see, the bioassay responses are much smaller and quite a number of them are negative. This is that antagonistic effect that I was discussing earlier. And so when we see a negative response like this, what it means is that there's some leftover testosterone or estradiol in the system, and the presence of the extract is actually suppressing that compared to the controls. Now, these bioassays were highly intercorrelated with each other, and this totally makes sense because the COX-2 and the IL-8 and the CYP1A1 are all mediated through the aryl hydrocarbon receptor. And even though these assays were done in different labs, the red shaded areas show us the strong, statistically significant positive correlation between the AhR-mediated assays in the two different labs. And then we see that there's a negative connection between many of the assays that are in red above and the estrogen receptor and androgen receptor assays. And this is because we see this antagonism effect. So now to get into the chemical results. So this is what an MS dial alignment looks like. What we're looking at in the middle, the series of green dots, those are our aligned features from our GC-EI data. And we've aligned them based on retention index to allow us to match to the NIST library based on both retention index and on mass spectral library match. Above that, you will see a bar chart which shows the presence, the abundance of that particular feature. So in this case, we're looking at a feature number 425, which is methyl mercaptobenzothiazole. And you see that that was present in the ash at much higher abundance than it was in either mix or mega, which are two of our standards, which contain a number of target compounds. So what that tells us is this compound was not one of our target compounds, and it wasn't present in the blank in any appreciable way. And so this is an evidence that this compound is real and found in the ash. On the bottom right, you see the EI mass spectral match, and you see what the retention index was on the top right. You see an EI similarity score on a scale of 1,000. So this is a pretty good match of a non-target feature. And so what we then do is we take the abundance of all these different aligned features and we look for how they're connected to bioassay responses. Before I go there, though, I want to just show you one of the quality control things we do with this GC-EI data. So we have a number of polycyclic aromatic hydrocarbons as target compounds in this analysis. And so on the y-axis of each of these 6 graphs is the area count we got out of the Agilent quantitative analysis program for known target compounds that are labeled, these 6 compounds that are labeled here. On the x-axis are the MS dial area counts for things that were automatically identified without telling the program that these were these compounds. It pulled these out and it identified them, and it came up with an area count that is strongly linearly related to the area count that we got from the quantitative analysis program. So you can think of the y-axis as representing the proper quantitation of these things, and you can think of the x-axis as the improper quantitation. But what it tells us is that when we have a non-target feature which has a particular area, that we expect that area count to be strongly related to the quantity of chemical in the sample. And that's important because it tells us that we can look for correlations between the abundances in the non-target data and the bioassay response. So this is hand waving to show that we can use these area counts in a quantitative way. So just to summarize all of the results that we got out of this, here I've shown a table which just counts the number of compounds which we pulled out of the data in various ways, and what happened after we filtered and what was related to bioassays. So the three streams of data are the GC electron ionization, the liquid chromatography with electrospray positive and electrospray negative. What you can see is that there were between hundreds and thousands of aligned features that were pulled out of that sample set. We do a series of quality control processes on those aligned features. We remove things with low signal to noise. We remove things that are present in the blank at an appreciable level. We remove things that aren't present in replicate injections in the case of LC. And then we, with that remaining set of features, which is still in the hundreds to thousands domain, what we're looking for is either a correlation between the response, and in this case, we're using Spearman rank order correlations between the bioassay responses and the abundance of a feature. What we see, just to focus in on the CYP1A1 line, just so you can understand what these numbers mean, we're looking at the GC-EI column, CYP1A1, and what that tells you is that there were 33 features out of the 407 that passed our filter that were positively correlated with cytochrome P450 activity at the 95% confidence level. And there were 11 features that were negatively correlated with the CYP1A1 response and at the 95% confidence level. And then the last number there, the one that's labeled EN, stands for elastic net regression. Elastic net regression is a technique which is a reduction technique. It's like a lasso regression, and you'll see it's pretty effective in identifying out of this kind of data where you have many, many descriptors in relatively few samples. It's a very good technique for identifying significant things. And so there were 13 things that the elastic net regression procedure picked out. And there's a significant overlap between the correlated features and the elastic net selected features. So for each of our 6 assays and each of our 3 instrumental platforms, you can see we have varying numbers of positive and negative correlations. And the elastic net regression in a number of cases returned no features, 0 features that were related to that particular bioassay endpoint. So you'll see in general, the elastic net procedure is stingier in assigning things, and later on you'll see that it has a better performance in terms of identifying things in final. So out of those hundreds to thousands of features, if we look at what's actually related to bioassays, we've got a much more manageable set between 76 and 276 compounds for the 3 different platforms. Now you'll see that there's many compounds that are higher in the samples that are residential than the ones that were in the undeveloped areas. But as we'll also see later, there's not a lot of connection between those compounds and the bioassay results that we looked at. So let's zoom in a little bit to think about those compounds which have a significant relation to bioassay results. Many of them, so here we're specifically looking at aryl hydrocarbon receptor binding results, and there were 76 GC compounds which had a statistically significant relationship to aryl hydrocarbon receptor activity. And you'll see that a lot of these things are not surprising in their ability to activate the aryl hydrocarbon receptor because they're aromatic hydrocarbons. And so lots of PAHs and things that are on our target list that we knew already would be significant in this regard showed up as significant. So, there's no new information content there, but on the other hand, if we already know all the things to look for, then we've satisfied ourselves in some way that we're not missing anything, right? But not all the suspects were quite so usual. So some of these are 3 compounds which were statistically related to bioassay response. They're also not, looking at their structures, not surprising that they might activate these receptors. These are compounds that were not in our standards, but for which we got good matches with retention index and MS, the spectra for the electron ionization in the GC. So we have many more of these. These are just 3 examples. So the real question, though, is how did we do in terms of finding compounds that might actually activate these receptors? Because the goal here is to fish out of complex environmental samples the compounds that matter. And there's lots of reasons why you think that might not work. Interactions between chemicals, mixture effects, inability to detect the right compounds using whatever ionization approach we've selected. So this table is kind of the summary of the work that we did, and I'm going to try to go through it a bit more slowly. So we saw, and here we're focusing on the GC EI data. And in this data, there were, remember, 407 features. And there's a database, Tox21, which you may be familiar with, which is an EPA hosted database, NIH hosted database that is an assessment of many, many different bioassay responses. And so there's several thousand compounds in Tox21 that have been run on the exact same CALUX aryl hydrocarbon receptor assay that we ran in this study. And so what we see is among the entire population of 407 compounds that were identified in the ASH samples and passed our various filter criteria, only 15 of those are known to be AhR agonists given the Tox21 results. So of our total detected universe, something less than 4% of the compounds activated the aryl hydrocarbon receptor. If we look instead only at the population that was statistically significantly correlated with the bioassay results, there were 4 of those compounds out of 17 that were correlated were in the Tox21 database and known to be AhR agonists. So that means 23% of the compounds among the compounds that were correlated were known to be actually active in the way that this assay is looking for. And then in ElasticNet, we found about the same performance. There were only 5 compounds returned by the ElasticNet procedure, and only 1 of those was in the Tox21 as a known agonist. But the problem here is the Tox21 coverage of our 407 compounds is not that great. And so we tried to expand our coverage. So the first thing we did is Mike Denison is the developer of this CALUX aryl hydrocarbon receptor assay that we're using, and his lab through previous NIH grants has surveyed over 300,000 compounds for aryl hydrocarbon receptor activity, and we recently assembled a database of those results, and there's something like 7,000 active compounds, which is much larger than the active compound set in Tox21. And so when we look at that database, the UCD database, we get a much higher fraction in all of the bins, but you can see that the performance goes up. But then the last thing we did was compounds that weren't in Tox21 and weren't in the UCDAVIS database were things that we could test ourselves. And so what we did is for the compounds that we still didn't know whether they were active or not, but they were correlated and we could get pure standards, we bought some pure standards and we tested them. And the estrogen receptor results here are similar, but we just have a much less significant database. And so the results look about the same as they did for Tox21 only, but we don't have this additional UCD database or the target tests for that piece. So I can't provide the last column of the table. So we went out and we bought 5 compounds and we tested them on the CALUX AhR assay. And what we found was that all 5 of them had statistically significant aryl hydrocarbon receptor agonist activity at different levels here. So you can see in the far right column, the prob value of the difference between the assay result and the blank, which is a solvent blank. And in all but 2 of these cases, these tests were statistically significant at the 95% level. And so what we confirmed was that this technique, this relationship of non-target features to bioassay results that we obtain on whole samples, these samples weren't fractionated in any way, they were just tested straight up on the assays. We used statistical procedures to fish things out, and as you see, we had a very high fraction of those compounds subsequently proven to be active on the receptor assay that we fished them out with. And so this suggests that we can indeed do what we're trying to do, which is test whole samples, use statistical procedures, find the unknown compounds, and identify them and confirm their presence in the samples, even when we have no prior information about the compound's biological activity in question. So that's been focused on the GC results, and frankly, we have the best and most comprehensive results for the GC-EI data because, and it's also probably a function of the aryl hydrocarbon receptor test is the place we have the best database, and the compounds that are in that that are active in the aryl hydrocarbon receptor tend to be GC-able in a way that the LC compounds aren't. But I wanted to spend at least a couple minutes talking about how we do the non-target in silico annotation using MS Finder. So we took our features, which were correlated with bioassay outcomes or were selected by the elastic net regression procedure, and we went back and we tried to do targeted MS-MS experiments on all of them. We got a fairly low coverage of successful targeted MS-MS experiments, however, because there's low abundance of some of these things, and it's hard to get enough fragment abundance. The other thing that's challenging is that many of these molecular formula are pretty generic. They're CHO or CNHO type compounds, which have many possible structures and not a lot of highly detailed fragmentation, and therefore they're not easy to nail down. But here I'm just providing one example of how MS Finder works. We've taken a particular feature out of our alignment that we know to be statistically important and related to the bioassay results, and we find that it has an empirical formula of C23H22O, and the structure that we think it is shown down in the lower right there, although this hasn't been confirmed, but this is the tentative match to that particular molecular formula given the in silico program. So just to give you a couple specific examples of things that are pulled out from the MS Finder exercise, these are 2 LC ESI negative on the left and 2 LC ESI positive compounds. The red spectral lines are the in silico predicted fragments, and the black lines are the accurate mass fragments measured in this MS-MS experiment that we performed. Between the inability to get a targeted MS-MS result for these samples, so there were many compounds we could not obtain a targeted MS-MS result for, and among the ones that we did get a targeted MS-MS, there was often not enough abundance to do very much with. So further efforts to concentrate, select, purify would be helpful here, and we're working on improving this set of the results. So where are we headed from here? First, we've proven that there are many things in urban wildfire samples that were not present in undeveloped samples, but based on the set of bioassays we chose to apply here, there's not a systematic difference that those residential samples are more bioactive. And we also did not see any systematic presence of the kinds of compounds that you would see in a typical environmental contaminants database. That is, after this fire, we don't see a lot of residue of known pesticides or known solvents. What we're seeing is highly converted compounds that have been, subject to combustion. I think we can say that in this project, the GC pathway yielded more success in isolating and annotating compounds that were related to the bioassay endpoints. And so for this purpose, it was more successful. That for other purposes, it may not be the same result, obviously. And then there's the fundamental question about whether we pick the right bioassays. There are many, many different biological endpoints that might be of concern, and it may be that the ones that we selected are not the ones that we most need to worry about. And that's the challenge for our toxicologist collaborators, to figure out what the right suite of assays is to apply to these kinds of samples. So I wanted to acknowledge the sources of funding for this work. We had two NIH grants that were focused on the fires. The Environmental Health Sciences Core Center at UCDAVIS provided some pilot funding for wildfire analysis, and an R21 grant from NIH also provided funding specifically to study this set of fires. So it was a rapid response grant following the 2017 fires. And as I mentioned, a lot of the methods that I talked about here were developed under the auspices of our Superfund Research Center that's cited here. I didn't list them on the authors, but as you'll see in a minute, they're an author on the paper that we recently published. The West Coast Metabolomics Center has been really helpful in helping my lab adapt and transform MS-Dial and MS-Finder for these kinds of applications. And in particular, Clay Blozis and Oliver Fien have been really critical to this effort. And so now I want to close by just making a plug. Just 2 days ago, our paper that covers the work that I talked about here was accepted for publication in Environmental Science and Technology. I can't tell you any more than that because I don't even have the proofs for the paper yet, but you should look for it shortly. The citation is below. It'll be out in a couple of days, possibly by the time this thing actually airs, because I'm recording it ahead of time. So I look forward to addressing any questions that you might have about this following the presentation. And I thank you for your attention and interest in the work.